The poster we have is actually, I think, on a very important topic, the one of sarcomatoid rhabdoid RCC. Now, sarcomatoid rhabdoid RCC is a very aggressive form of renal cell carcinoma that can occur on a backbone of either clear cell RCC or non-clear cell RCC. So this is a form of disease that has unfortunately not benefited from the revolution that we saw with target therapy. So although patients with metastatic disease, particularly clear cell RCC, have benefited tremendously from target therapies, patients with sarcomatoid and rhabdoid features still seem to be doing poorly and did not respond a lot to target therapies such as sunitinib, sorafenib, and other tyrosine kinase inhibitors. Now, what is very interesting is that recently the immune checkpoint inhibitor-based trials, such as the pembro-axi trial, the pembrolizumab axitinib trial, the avelumab axitinib trial, the nivolumab ipilimumab trial, and even the atezolizumab bevacizumab trial, have all shown within the subgroup of patients with sarcomatoid RCC that there seems to be a distinct benefit within that subgroup. Now, this is still preliminary data that has not been published in journals yet, but has been presented at meetings, and that is suggesting the strong signal. So what we set out to do is to ask, is the signal real? We tried to pull multiple different data sets. We looked at, first of all, the Harvard data set. So this is regrouping patients from Dana-Farber Cancer Center, from MGH, and from BIDMC, and we asked whether either monotherapy or combination-based therapy immune checkpoint inhibitors are favorable for patients with sarcomatoid and rhabdoid RCC. We did the same in a very large IMDC data set that includes more than 40 centers across the world, and we also looked at a subgroup analysis, actually pooled analysis of two clinical trials, the Checkmate 010 and Checkmate 025 trial of nivolumab monotherapy, and we found in all of these that immune checkpoint inhibitor seems to be conferring a strong benefit to patients with either sarcomatoid and or rhabdoid RCC. So we know that they're responding very well, so we tried to ask, what are the molecular drivers of disease? We know very little about what is happening in disease, what are the molecular characteristics of disease. So we first looked at the DNA level. What we found was that these tumors harbor mutations in genes such as NF2, BAP1 that are actually associated usually with a poor prognosis, but also deletions in CDK and in the CDKN2A locus and amplifications in the EZH2 locus. Now, I think this is very important because this, first of all, gets at the question of why these patients have a poor prognosis. These mutations in these genes have been associated with poor prognosis, but second, it opens the door towards targeting the pathways that these genes regulate. So, for instance, NF2 mutations could lead to upregulations of the APTAS pathway, which could potentially be targeted. The CDKN2A deletion could lead to sensitivity to CDK4/6 inhibitors. EZH2 amplifications could lend itself to targeting by EZH2 inhibitors. Now, this is all still just based on the molecular data. Unfortunately, we still do not have clinical data to support this, but I think this is all very promising. Now, then we asked at the RNA level, what is happening in these tumors? What we found is that actually the RNA level data is much more marked at the DNA level. The signal was much greater than the DNA level, and that they had a very distinctive transcriptional signature. This was characterized by things like a high cell proliferation signature, such as E2F and G2M, but also a very immune inflamed phenotype with pathways such as IL-6, JAK-STAT3, and IL-2, STAT5 signaling being upregulated. I think the importance, and also a very strong mixed signature. The importance of this is twofold. One, in particular, the mixed signatures seem to be predicting poor outcomes within that subgroup of sarcomatoid and rhabdoid RCC. So we have a hint of what is driving the poor prognosis of these tumors at the molecular level, and also from an immune perspective, these tumors seem to be very immune inflamed, which may be driving the immune checkpoint responsiveness. We also found, using IHC on more than 800, or I think 600 tumors, we found that sarcomatoid and rhabdoid RCC tumors seem to have higher PD-L1 expression at the protein level, but also seem to have higher infiltration of CD8s within the tumor, which all would potentially associate with immune checkpoint responsiveness. So I think, in conclusion, that these tumors are responding very well to immune checkpoint inhibitor, and I think we've presented the most extensive data on that to show that. And I think these tumors, in the absence of better alternatives, should be receiving probably these regimens. A second important point is that these tumors have distinctive molecular features, both at the DNA and the RNA level, and this might explain both their historical poor prognosis, but also their responsiveness to immune checkpoint inhibitors.